Great. Well, thanks for the introduction and thanks for the patience while we figure out how to make hybrid presentations work properly. So as Eleanor mentioned, I'm now an assistant professor at UMass Medical School. I was a postdoc at MIT Biology before this and a grad student at the University of Chicago. And my trajectory has taken, it's all about RNA, but taken various different forms over the last 10 to 12 years or so. So I'm going to give you a little bit of some insight in some collaborative work that I did when I was a postdoc that really kind of sets the stage for why my lab is interested in what we call the efficiency of creating mRNA molecules. And we're very interested in it from a mechanistic perspective, but as I'll try to sprinkle throughout the talk, there are some consequences for understanding how our cells and our bodies and we as organisms actually are able to temporally respond to various stimuli, conditions, environments, diseases, and so on and so forth. Okay. So as a grad student, and I still am, I've been very interested for my entire career in how genetic information and specifically nucleotide sequences that are found in DNA can translate all the way through to create a diversity of cellular states and phenotypes. And what my career has taken a path towards is actually trying to say, well, let's actually start to understand some of the molecular mechanisms and the actual regulatory molecules that might underlie how that information gets carried from the DNA sequence all the way to enacting a different cellular state or phenotype. So as I mentioned, the place that I've really focused on for the last 5, well, maybe 9 years now is only on RNA. You know, we often think of as biologists, the central dogma is kind of this like big steps that happens. You go from DNA to RNA and RNA to protein and everything in the middle is transcription, and that's what we call it. Turns out it's obviously much, much more complex than that, where the process of creating an RNA molecule, is this working? Yeah, okay, involves not just actually transcribing that molecule, but also processing or splicing and cleaving that molecule. And that's what I'm going to talk a little bit about today, but also efficiently exporting RNA to the cytoplasm, localizing it to the right place, and then a whole host of mechanisms involved in translation that my lab does not really think about very much. So the other complexity to this is that we only have a certain amount of genes in any of our cells, but those set number of genes actually give rise to potentially even an order of magnitude more different RNA molecules that can be produced. And these are called isoforms, and where these different RNA isoforms are created by different alternative splicing or 3' end or even start type decisions. So what we study and what I'm going to tell you about a little bit about today is how can we actually understand the processes that give rise to different RNA isoforms, and what are the different ways that RNA isoforms can be regulated in different conditions. Okay, so just to give you a quick primer on the mechanisms behind splicing. So splicing is an essential step in the mRNA life cycle. In mammalian cells, greater than 95% of genes are actually spliced, and that means that they have an intron intervening to exonic sequences that eventually code for protein. And this intron is actually excised in a 2-step catalytic reaction, the details of which I might go into a tiny bit more a little bit later in the talk. So the act of actually splicing a molecule can result in multiple different exons being stitched together. And greater than 90% of mammalian genes can actually conduct this process in alternative fashion. So you can have alternative splicing that can result in different exons being included, different start sites or end sites, and these can have an impact on either protein composition through different coding exons being involved, or mRNA levels, localization, and general regulation through a whole host of either coding exons or non-coding exon regulation. Splicing is very cell type and tissue specifically regulated, and we know quite a bit about the molecules that actually regulate splicing, but we know much less about how splicing is dynamically regulated in different conditions. So I think that I don't need to go into too much detail here about this, because I believe you had a workshop this morning that I couldn't attend on the details of RNA processing, but I want to kind of highlight a couple things that are crucial for understanding how to measure splicing differences and RNA isoform differences with RNA sequencing data. So from a pool of RNA, much of the data I'm going to talk about in the first part of my talk is polyadenylated RNA sequencing data, which means that we're able to capture a mature mRNA molecule by using a hook on this poly A tail, using an oligo DT, and using a biotin streptavidin interaction to pull that down. We can then select that poly A, get mRNA, fragment the mRNA, make into DNA fragments, sequence all those millions of fragments, and then map the reads. So the crucial part for understanding splicing is that not only do we just map those reads to different exons and different genes and different exons, but we can also actually use a subset of those reads to understand how exons are connected together after splicing occurs. And that's through these reads called splicing junction reads that are quite critical to understand whether or not either these two exons are connected together, as indicated by this top read, or if all three of these exons are all in the same isoform, and that's what these three bottom reads represent. Okay. So I'm going to go into the biology portion using all RNA sequencing data and tell you two quick stories. The first about RNA processing and RNA splicing dynamics in the human immune response, and the second about actually measuring the rates and modeling the kinetics of mRNA splicing, and that's work that we did in flies. Okay, so the first question I want to ask, and this was a question that we were very interested in, is how is mRNA processing dynamically regulated in response to an external stimulus, rather than how our cells are normally creating mRNA on a homeostatic fashion on a minute-by-minute basis. So the place that we went to look at this was an ancient and very, very conserved human response, and that is the innate immune response, where cells like macrophages are primed to respond to external pathogens very, very quickly and very rapidly. And here's a electron micrograph of a macrophage reaching out and engulfing E. coli cells. And so when macrophages or other immune cell types are infected with any sort of pathogens, there's a very quick and rapid response that involves the creation and kind of propagation of cytokines that are used as signals to tell other cells and recruit other helper cells. And I'm not really going to get into the immune biology here, but one thing that was really interesting when you read this literature is that people know that a lot of these responses are actually initially triggered by a cascade of transcriptional responses where different mRNAs are produced in order to start making these signal to peptides and actually enacting the immune response in an early fashion. And one thing that was quite fascinating is that especially in very early stages of the innate immune response in mammals, you actually not only have specific mRNAs that are differentially transcribed, you also have a huge increase in the total amount of mRNA that each cell makes. And so now this is suggesting that there's a huge global upregulation of mRNA, and we wanted to understand, well, what are the global effects and what might actually be regulating more fine-tuned responses of specific genes starting to be more upregulated or perhaps not as much and just be kind of carried along with the global effects. Okay, so in order to do this, I collaborated with Luis Barrero, who is then at the University of Montreal and is now at University of Chicago. And I believe Rachel probably knows him fairly well. What we did was we collected blood from 60 individuals, extracted macrophages, and then infected these macrophages with two different bacteria, Listeria and Salmonella, independently. And then we took these infected macrophages from 60 individuals with both non-treated and two different treatment conditions, and conducted RNA-seq after both 2 and 24 hours of infection. And we did RNA-seq in order to measure three different things. The first is mRNA expression, so gene expression. The second is to actually look at splicing responses and mRNA isoform levels. And the last is we conducted small RNA-seq to start to look at some of the regulatory RNAs, especially microRNAs that might be involved. So we see a whole host of changes in gene expression and isoform usage, none of which is surprising. As I mentioned, we knew that mRNA was hugely differentially expressed after the immune response. And so we see that 30 to 45% of genes are differentially expressed. We also see a large change in mRNA isoform usage. And so now this is on the level of entire isoforms that might be up or down regulated. And we see that 13 to 25% of genes have differential isoform usage. What's really interesting is that when we look at the overlap between the two, we do see a large degree of overlap between differentially expressed and differentially spliced genes, but we see that as much as 10% of genes only show changes in isoform usage. And that was quite interesting to us because it suggested that these are non-overlapping regulatory pathways that are actually used perhaps to do what I was talking about before and fine-tune global or more specific gene regulatory responses and create specific isoforms that are necessary independent of whether or not you need more or less of that gene to be expressed. Very reassuringly, both sets of these genes were enriched in immune-related genes, and so we knew that these were, from a pathway perspective, likely actually respond in a involved in a final response. Okay, so to understand the regulation of these, we went in and actually started to ask about specific exons rather than a whole isoform level or whole gene expression level that could change. And we found many different changes. I'm going to focus only on two today. The first is alternative splice sites, and that is that we saw the differential inclusion of different cassette exons that are either included or entirely excluded from a final isoform. And the second is alternative polydenylation sites, and this governs the composition of the 3' end of a gene, and specifically the differences in 3' UTR or untranslated regions, either by using an entirely different exon at the very end or using a different cleavage site at the very end of the same exon. And so before I get into the results, I want to tell you how we actually measure splicing differences. And this is through a metric we refer to as % spliced in, or psi values. And very simply, this is essentially using the number of reads that support an inclusion isoform, which in this case is the inclusion of this exon in the isoform, versus the number of reads, and these are junction reads, that support the exclusion isoform. And we can calculate a proportion of the % that are actually supporting the inclusion isoform, and then ask, how does that % change after treatment or across different samples? Okay, so what I'm showing you here is a distribution of isoform changes across these skipped exon categories and this 3' UTR change, and specifically tandem 3' UTRs. And what we actually see is we see a global, we not only see thousands of specific changes, but we actually see a global shift towards increased inclusion of cassette exons across hundreds of different genes, and a global shift towards the usage of shorter 3' UTRs in general across, and so this is more than 80% of genes now use a shorter form of the 3' UTR after infection. Okay, and so this is kind of generally what it looks like. We have a shorter 3' UTR, but more internal exons. We see that this exon and 3' UTR regulation are very general properties. We say that they're persistent across time. This is a delta psi across 2 hours versus 24 hours, and you see that skipped exons and the 3' UTR changes are consistent, while other exon types are actually more dynamic and change between early or later infection. And we also see that these responses are evolutionarily conserved. I'm now showing you the same mean delta psi for 4 hours of infection. Now, this is whole blood infected with LPS, and there's a whole host of other experimental considerations, but this is showing the conservation of the specific genes between human, chimpanzee, and macaque. And we see the same global shifts where we tend to see more skipped exon inclusion and shorter 3' UTRs used across species in the same sets of genes. Okay, so why are 3' UTRs shortened upon infection? This is where we get to asking the actual mechanistic question of what's actually happening in the cells when we see this very global pattern. So 3' UTRs are used for regulation of many of mRNAs in many, many different ways, but one of the primary ways that people have thought about them is as a binding site for these microRNA target sites. And microRNAs, again, can also do many things, but one of the primary things they're taught to do is actually bind to this 3' UTR and target this mRNA transcript for degradation through binding of a RISC complex. In the end, you're likely to have a downregulation of mRNAs that are bound by microRNAs. So what we did was we asked, well, can we actually see more microRNA binding sites in these longer regions that are lost upon immune infection? And not only that, can we see specific microRNAs that are actually enriched within those sites? So we calculated an enrichment score, and we see not only specific microRNAs, these microRNAs are all microRNAs that are upregulated upon infection, but we also see a significant overlap between the microRNAs that are enriched in those sites for the 2 different bacteria. And so what this led us to hypothesize through a whole series of other analyses is that 3' UTRs upon immune response seem to be specifically shortened in order to allow a gene to continue to be upregulated independent of microRNAs that might come in other contexts and actually target that mRNA for downregulation. So this was very interesting to us from both the mechanistic perspective, but also a dynamic perspective, because we realized we can start to use these data to understand what is the temporal regulation of various different cellular conditions. And so this is by no means specific to immune response. This is a figure showing global shifts that have been seen across various different portions of an mRNA and in different environmental stimulation or disease conditions. And so one thing that we were very interested in was, well, why are these global shifts, right? We do know quite a bit about each specific pathways that are enriched in each of these different contexts, but what's happening globally that could cause the cell to say, well, I'm just going to enact a huge whole-scale change of the portion of an mRNA that's actually regulated. And so this is getting into stuff that we actually study in my lab, where we wanted to actually understand, can we go in and ask, not just from a steady state perspective, which is to look at these kind of broad steady state levels of mRNA, but actually look from a temporal perspective. Can we ask about the rate at which each of these changes are regulated? And can we ask about the rate at which mRNAs are produced? And one of the initial questions we're aiming to answer in my lab is, can we actually determine the rate limiting step for creating an mRNA, a different mRNA isoform across different genes, and use this to understand whether or not the global targeting of these different processes in different environmental or stimulation or disease contexts can be understood from a mechanistic perspective better. So I'm going to tell you very quickly about how we've done this, especially for splicing, and how we think that this provides quite a bit of promise for doing this for other mechanisms. Okay, so RNA sequencing captures mature and stable mRNA, and that's through the use of that poly-A tail. But in order to actually understand the temporal dynamics of it, we now have to start to look at intermediates in the same way that if you're doing a stimulation, you want to have a time course across different conditions to see what the trajectory is. So the way in which we actually capture these intermediates is by a technique called metabolic labeling, where we incorporate a 4-thiouridine, which is a synthetic modification of a normal uracil base, into cells for a short amount of time. We're then able to selectively biotinylate these labeled molecules and do a pull down that allows us to sequence only the nascent or very newly created mRNA molecules that are likely to be enriched with various splicing intermediates, including both things that have been spliced, but also unspliced. And we can see over time how fast do they get spliced. And then we can take these and create RNA sequencing libraries and sequence them to get millions of short reads. So we did this in Drosophila, and this was, and oops, let me forward a little bit, in collaboration with Karen Edelman, who's then at NIHS and now is at Harvard, where we looked at a time course of metabolic labeling after 5, 10, and 20 minutes. And what we can see reassuringly is that we see greater numbers of introns being spliced out with increasing labeling times. And that's exactly what we expect because they've had more time to be sliced out. And what we wanted to know is, well, what is the actual rate of intron loss, and how and we can use that as a metric to measure the splicing rate of a particular intron. Okay. So I know there are some people who think about modeling in this audience. So I'm going to give you a little bit of a teaser into how RNA-seq data is not super straightforward to think of when you start playing with the protocols quite a bit. Because what I showed you before is kind of nice, right? We can take a time course, we can fit an exponential curve, which is what we expect a half-life to be, and get a half-life. But because of the experiment, it's not that simple. Because that plot I showed you assumes that the intron was created at exactly time 0 and it's had the exact labeling time to be spliced out. But that's not true because of how we do the experiment. Instead, it's had the entire, say, 5 or 10 minutes to actually be spliced out, and it could have actually initially been transcribed somewhere in the middle of that period, so it has a much shorter lifetime to be spliced out. And there's other considerations of the fact that it could have been created before or during that actual labeling time, and that's a problem. So what we want to do is we actually want to use these spliced exon-exon junction reads or unspliced reads, which are kind of traversing the intronic exonic boundary, to assign them to different lifetimes and say, okay, if we knew which of these reads had what amount of time to actually be spliced out, we could very easily solve this problem and say, all right, here's the splicing life, and we can half-life, and we can assign an exponential curve to it. But of course, our data looks like what's on the bottom. And so without going into the math, what this necessitated is that we actually had to derive a probabilistic model that allows us to look at the distribution of lifetimes across the distribution of lifetimes we thought we had in the data, and use that to understand what's the probability of seeing a spliced or unspliced molecule given a particular half-life. This is published work. I'm happy to answer any questions on the actual way in which we did that. But what we were able to use that to do is actually measure the rate of splicing on an intron specific level. And what I'm showing you here are two different actual coverage plots to show you that we can use these data to do that type of measurement. Darker colors are earlier time points, and lighter colors in both purple and gray are later time points. And what you can see across this is indeed that you start to lose out more of the intron as time goes on. On the right, I'm showing you that just with those junction reads that are actually specifically informative, seeing more exon-exon junction or more spliced reads and fewer intron-exon or unspliced reads over time. And this is specifically what we're using to model it. Okay, so what we wanted to use these data for is actually to ask about what underlies variability in the rates of splicing. Can we start to understand something about the mechanism before we apply this to all sorts of fun conditions? Okay, so we calculated intron half-lives in Drosophila for a little bit more than 25,000 different introns, and we find that the median rate of actually excising this intron is a little bit less than 2 minutes. We wanted to know, well, what's actually underlying the splicing out? And one of the first things we thought about was, well, maybe it's the time it takes to pair together the 5' splice site with this branch point. That's the very first reaction in that catalytic cycle that initiates splicing. So one of the obvious things is, well, maybe what underlies the rate of bringing these sites together is just the length of an intron. The longer it is, the more time it takes to bring together those sites, the shorter, the faster it is. And this is what this looks like. This is intron length versus the half-life of introns on the y-axis, where each dot is a single intron. And that blue line is a running median that shows you just the average across the length. And we don't really see a strong correlation, but what we do see is a very, very predominant dip right at 60 nucleotides. We wondered, well, what is that? So one clue was that in Drosophila specifically, the mode of intron length is actually 65 nucleotides, and greater than 30% of introns fall within this 60 to 70 nucleotide length range. And it turns out in Drosophila, with all of the transpositions and kind of jumping around of different repeat elements going on, there's selective pressures that seem to favor shorter genomes and shorter introns. And so when we zoom in on this, we see that exactly at 60 to 70 nucleotides is exactly where you have the fastest splicing rates. The rates get slightly slower with longer introns, as you might expect. And interestingly enough, they actually also get slower with shorter introns. And through some other analyses, we realized that this is probably because if you have a very, very short intron, you don't have enough sequence space to put all the elements you need to conduct the molecular reaction. And so you run into a steric boundary to how you can fit everything in there. So what was fascinating about this for me is that we can actually start to understand something about the design from an evolutionary perspective of how much can you put into introns. And in another story that I'm not going to have at all time to tell you about today, we actually tried to understand, well, how are introns that are very, very long spliced out in Drosophila if there's this huge pressure, both from an efficiency perspective, but also an evolutionary perspective to get shorter introns. And we realized that introns that are very, very long and need to be maintained as such for other reasons, because they harbor other elements or in particular genes that need long introns, have other ways of efficiently splicing out their introns. Okay. Lastly, what we saw that was quite fascinating is that introns that are involved in these alternative splicing decisions tend to actually be spliced out slower, independent of their actual length. And that suggested to us that indeed there is a mechanistic efficiency or kinetic process that goes on in determining whether or not alternative splicing decisions that are probably very likely important in the context of our cells responding to different contexts are actually regulated from a temporal kinetic fashion. And what's really exciting is that from new data, and this is unpublished, the only slide that's unpublished, but we can also start to measure the rate of 3' end cleavage at the very 3' end of a gene. And so we can look again at whether or not genes that have alternative 3' end decisions have different kinetics than genes that don't have a difference and only make one decision. And what we see on the right-hand plot here is that for genes that have alternative 3' end sites, very similarly to splice sites, they actually show slower cleavage and polynylation than genes that have a single transcription end site. So this again suggests to us that there is indeed likely something that's regulating from a kinetic and mechanistic perspective the rate at which these decisions are actually being made. So I'll end by saying that what we're trying to now do is apply these methods to various different specific conditions. And so just on this list, we're looking at immune stimulation in cancer, trying to understand whether in these contexts there is indeed pressures to kinetically or from an efficiency perspective, regulate the extent to which you can create different mRNA isoforms and what consequences that has. And by thanking many people in my lab and the collaborators for the work that I talked about today are Luis Barrera in his lab, Karen Edelman in her lab, and Chris Burge in his lab. Thank you very much.